Good afternoon, everyone, to our audience offline and also online. We welcome you to our seminar series. So today we have the pleasure of having Dr. Sarkar with us here today, and I would request Dr. Algebrabased to kindly introduce him.
Well, thank you, Alpana, for organizing this. And thank you, Navonil, for coming here and agreeing to give this talk. So Navonil has done his BSc in zoology and his MSc in environmental sciences. After that, he did his PhD at the Indian Statistical Institute. That is where we met. Many of us met Navonil. Then Navonil went to the US for a postdoc at the Fred Hutchinson Cancer Center in Seattle, where he did his postdoc, a very successful stint as a postdoc, and then eventually he was observed as a staff scientist there. Navonil has very recently joined the Department of Pathology at the Medical College of Wisconsin. So he has come here for his visit to his parental home, and we have grabbed this opportunity to get him here. So thank you, Navonil. Without further ado, let's start.
Thank you again for this opportunity to speak in front of all of you. I remember I was here in 2018, March, presenting last iteration of presentation. At that point, I was working on a tool that accurately determining DNA repair deficiency. And at the very end, my last slide was something that I promised that I'm working on Cellfree DNA, and it would be inferring something that is beyond genomics, like not that something as a DNA sequence or mutations or copy number alterations, but beyond that. It took several years to formalize that study. And finally, we came up with this work. And very recently it got accepted. It's getting ready for a publication. And we will show that how we are inferring or we are extracting way more information than the mutation or copy number with the variations from cell-free DNA. So that is exactly what is today's talk is all about. Now, as you can imagine, like this is, you can imagine from this band that the author numbers are not small, and everybody is contributed in a big way. But the major contribution is from Robert D. Patton, Gavin Ha, and Pete, and me, obviously. So all four of us as team together worked really hard to kind of come up with this, I would say, discovery that we have made with cell-free DNA. Now, before diving into the details of the cell-free DNA science, we have executed or implemented all our studies in the setup of prostate cancer. So we will briefly introduce what prostate cancer is and what are the kind of disease progression trajectories prostate cancer patients usually go through. So prostate cancer usually appears as an adenocarcinoma, which stays localized. And at that point, the most of the therapy options are like surgery or radiation therapy, and it seems to be working for majority of the cases. 90% of the patients, this is the cure, surgery or radiation therapy. So it's not a problem. Like they can survive 15, 20 years fine. But the other 10% of the cases start showing resistance to this initial treatment, and the tumor sites start growing. At that point, the alternative approach come up, which is androgen deprivation therapy, which is basically chemical ablation of androgen hormone, which is the primary driver of prostate cancer at the very beginning. So this androgen deprivation therapy also seems to be working for almost 100% of the cases, but inevitably the disease becomes resistant to this treatment option, and the tumor starts growing back again. And at that point, it usually takes the form which we call metastatic disease. We all know the metastatic means it is no more localized. it has actually segregated to different parts of the body. It starts affecting the various other organs. So that's the metastatic form. And in this case, there are no practically curative or effective treatment options left. People try various generic chemo, but the overall effect or response is really short. Like for a month, 2 months, 3 months, it's substantial. It's been considered, and the ultimate fate is disease-related death. So it has very recently recognized, if I'm saying this recent, but still it is 5-6 years back, people started seeing this very clearly, that this post-ADT stage, prostate cancer is no more just adenocarcinoma. Various phenotypic subtypes are start showing up. And some of them are named as neuroendocrine prostate cancer, double negative, amphicrine, AR low, likewise. So these are different names. Just we have to imagine that these names has a molecular basis. Like each of these names has a molecular basis. They are no more regulated under androgen, but some other transcription factor became activated, and they have actually accepted various lineage trajectories, and the disease start progressing in that form. The challenge part is genomically or DNA sequence wise, adenocarcinoma and these progressed various phenotypic subtypes, they are very similar. Not very many mutations occurred in between these two stages when they were adenocarcinoma and when they became castration-resistant prostate cancer. But what has occurred is the transcription program, it has taken a very different trajectory than the initial one. So if we have a sample that we have collected at this stage or during this surgery, they might talk about those DNA mutations or copy number aberrations that has been the driving role for this disease state. But we might not capture all those epigenetic alterations that has happened in this stage, and that's how the later treatment options based off of this primary tumor might not be the most accurate way of dealing with this disease. So yeah, here I'm showing a cartoon is how this was an adenocarcinoma. Then there's one cell that's tend to become a kind of adopted a new transcription program, and they became more selective or favorable during this ADT or androgen deprivation therapy. Now onwards, I will call ADT, all these androgen anti-androgen therapies. And then eventually those lineage plastic adenocarcinoma, transformed adenocarcinomas becomes the primary one, and all of the cells become NEPCs. Why all of them become NEPCs? Because ADTs stop growing as you are putting them on the anti-androgen therapy, but this NEPC do not respond to those deprivation, and they are very aggressive. Maybe 10 times or 20 times. I don't have exact estimate how more aggressive it is, but their cell cycle progression rate is substantially higher than adenocarcinoma. And they can kind of take over very quickly the entire tumor. So here I'm showing an example. This is the adenocarcinoma slides or histopathology slides, and this is the neuroendocrine prostate cancer histopathology slides. They have a characteristics difference. Like these cells are substantially bigger. My apologies. I mean, I was not aware of the overall resolutions that I would get, but here the resolution is pretty low. So these cell sizes are smaller. Their overall nucleus sizes are small, and it has a more cytoplasm here. Whereas here, the entire cell volume is taken up by nucleus and not much cytoplasm left. And overall cell size is also small. So it is a small cell type. And when we take these examples from the actual patients and did perform a transcriptome or total transcriptome analysis, what we found out is there are some androgen regulated gene sets that includes also AR gene itself or androgen receptor gene itself. They are highly expressed in adenocarcinoma, but those genes expressions are lower in NEPC because they are not dependent on those. And there are some neuroendocrine prostate cancer specific genes or neuroendocrine marker genes, they are highly expressed in NEPCs or neuroendocrine prostate cancers, and those highly expressed in NEPC, but not so much expression in androgen-regulated prostate cancer. So you can clearly see that transcription program-wise, they can be separated from each other very easily. But the practical challenge is how this has, we know this from some selective patients, one-time sampling. But how this would be informative for a patient who is undergoing anti-androgen therapy? Can we infer that when in their kind of course of disease progression, it becomes neuroendocrine prostate cancer? Can we really predict that? We have no way. I mean, you cannot go on doing a repeated biopsy over the time on a single patient. That's not feasible. And the prostate biopsy itself is a tremendous task. It has to go through some anal pore, and the camera has to go, and it's a huge process. It's not only costly, it has lots of comorbidities associated with the process itself. And imagine somebody got a metastasis, like the tumors is all over the places, and maybe one tumor become neuroendocrine. Can we detect that? Then we have to kind of sample every single pieces of it and then run histopathology or perform RNA-seq, so it's not practical. So we needed some practical solutions for this. And at that point, I was working on this cell-free DNA assays, and I thought that this might be a non-invasive approach, and we probably can use cell-free approaches to determine this phenotypic subtype in patients and in the real-life patient samples. So at that point, when I started working on ctDNA, that is in 2017, at that point, mostly what we were doing was using cell-free DNA to kind of call mutations, copy number changes. Those were the standard of care, like standard information derivations that was possible, and then that was it. But then we realized that for prostate cancer, it might not be that much of informative. What we need is beyond that mutations barrier. Like we need to have an ability to predict mRNA expression. Then people start talking about analyzing cell-free RNA, and cell-free RNA is terrible quality-wise and quantity-wise overall. So and practically it was not even representative of the tumors at all. So we thought that is also not a great idea. So we thought that, yes, I mean, we have this cell-free DNA and we have to come up with a new approach from these new approaches. And what we could do is studying DNA, but have an ability to predict their mRNA expression and having their ability to predict their epigenome. So can we do that? So that is the kind of hypothesis that we set up, is cancer phenotype, that epigenetic phenotype, can be determined through cell-free DNA analysis. That was our hypothesis that at that point we started our work on. Now I will show you a cell-free DNA versus regular DNA whole genome sequence IGV map. I think most of you have already seen what is IGV map of a sequence data or NGS data. Do you? All of you, do you know that? So this is the gray color is a regular fragmented whole genome sequence data, which is actually showing some random pattern here, pattern of coverage. Like this is the one, what we are showing here is ASCL1. This is the one gene body. And here what we are showing is about, say, 70x depth of coverage of sequencing whole genome. And that has some randomness in terms of the coverage profile. Whereas this violet color, which is basically the cell, it's derived from a cell-free DNA whole genome, when we align, we started seeing a kind of peakedness or peak and deep pattern. And we have also seen that when we are isolating cell-free DNA from a patient, it has a typical size classes. The primary cell-free DNA fragments are 167 nucleotide, that is the kind of modal class. Then there is a second peak, which is around 350 or 330, 340 nucleotide. That is the second peak. And then there is a third peak, which is roughly around 450-ish. So we started thinking back that why this fragment size has a specific class, why when we are aligning, it has a peak and dip. And it took us a few months to come up with this thought, and many others has also came up with the same idea at that point. And we discussed these are basically the primary peak is mononucleosomal fragments. So if you have a nucleosomes and DNA is wrapped around, which is basically an 167 nucleotide long fragments. And then there is a linker space of 20 nucleotide. Then again, you have a second, another nucleosomes. Now imagine if the fragments happen, like it retains 2 nucleosomes and fragments happens on the either side of those two, then that would create that 330, 340 nucleotide long fragments, then the trinucleosome fragments. So basically from cell-free DNA, what we were seeing is mononucleosome, dinucleosome or trinucleosome fragments. So this was our initial library prep derived observation. But then we immediately go back and started talking to the molecular biologist. And actually, he's been more than that, Stephen Hanikoff. He was in Fred Hutchinson Cancer Center. And he said that he's working on an assay that can accurately, which is the alternative or orthogonal alternative of ChIP-seq, where they are using MNase enzyme to kind of cut this genomic DNA along the sites wherever it is not protected by some DNA-bound protein. And in his assay, he's also seeing subnucleosomal fragments, which are shorter than this 167, but those nucleotide fragments are not seen in our cell-free DNA. So this question becomes like, why? Then we go back and start working again, and we perfected our library prep approach. So our initial library prep was not right. That's why we were losing those smaller fragments. Then as we worked up, we also started capturing the smaller fragments. Now, when we started smaller fragments, then we know that those smaller fragments are subnucleosomal, which is basically transcription factors, polymerases, that has smaller profile on the DNA. So those are creating smaller fragments.
What is the sample for your cell-free?
At this point, these are the two tumors derived from PDX.
The tumor sample itself is?
So we have the tumor, and we have the plasma collected from the PDX mouse.
The plasma is the sample for the cell-free?
So plasma is the sample for cell-free. Yeah. So here onwards, so what we did is now we know that what we are seeing, this peak and dip, is not a random peak and dip. It has some biology, and we have to come up with a formal approach, formal computational methods that would give us or extract all these informations, and we can reliably potentially use those to infer epigenetic variants from the different cancer types. So what we did, we have access to a PDX score in Fred Hutchinson Cancer Research Center and University of Washington. They were maintaining total 46 PDX lines, prostate cancer PDX lines. Of those 46, we had access to roughly about 25 of them. And among those 25, 18 were adenocarcinoma, 6 were neuroendocrine prostate cancer, and 2 were AR low phenotypic subtype of prostate cancer. So we already got what we needed. We thought that PDX is the great model to start with because we could have started with the patient samples, but PDX is better because if we start with the human samples, they have a different levels of normal as a background. And when we will train our computation model, those background will create lots of problems. Whereas when we are starting with this PDX mouse, here, all those fragmented DNA that are present in the plasma is coming from the tumor, as well as the normal sections is coming from the mouse. And we have the ability to bioinformatically subtract this mouse easily, and then we have 100% pure human cell-free DNA. So the model cell-free DNA could be derived from them. So that was our start point. And when we are generating this cell-free DNA side by side, what we could do is we could also run ataxic on those tumors, same lines from the same passage, and we could also run some chip6 orthogonal approach, which is cut and run in this case, that Steve Henikoff has generated. And we actually look for these marks, H3K27 acetyl, H3K4 monomethyl, and H3K27 trimethyl. These are very common epigenetic mark, which talks about the genes like activity to some extent, like gene promoter activity to some extent, very grossly speaking. So that's what we do is we started with this mouse plasma, we perform whole genome sequence, subtract mouse, and then we get the pure human circulating tumor DNA samples, and side by side from the same PDX mouse we perform metaxic and cut and run, and we have this overall chromatin profile side by side. So this is the kind of first kind of such data that we have side by side, right from the tumor and what we derive from the plasma. And then what we did is we train our model and then gone back and try to learn that, okay, so is this all the genomic features or can we learn more? So kind of we gone to and fro, up and down, like starting from here, we generated a model and applied it in the real life patient samples, and then again derived our experience, gone back and retrain our model, and that's how we came up with several usable features that I will show in a few slides from now. So before again diving deeper into it, I thought that because here is a mixed population, many of you cell-free DNA, the concept of cell-free DNA might be new. So I'm showing this this particular slide for you all. So cell-free DNA is generated when a cell is apoptosizing or dying. And when the cell is dead, at that point, caspase-3 or caspase-activated DNase enzymes get activated, and it starts chopping off all those DNAs like as much as possible. And it could show maximum activity to the DNA regions that are not protected by any DNA-bound protein. So basically what it is happening here is, so this is the nucleosome protected region, this is nucleosome protected region, this is transcription factor protected region. These particular regions on DNA stays longer in the system, whereas the regions that are exposed, they are chopped off or degraded faster. And that's how these fragments get released, and that forms the cell-free DNA. Now, imagine when somebody don't have a cancer, the entire cell-free DNA pool would be coming from the different normal cells. But if somebody has a cancer is maybe smaller part of the body, but its overall turnover, the number of cell cycles and number of cells dying is also higher. So it would also form and contribute to this overall cell-free DNA burden. So here what we are terming it is the proportion that is coming from the tumor is circulating tumor DNA or ctDNA, and then there will be circulating normal DNA that is coming from the normal part, and overall it forms cell-free DNA pool. So that is how we are defining. And as I just explained to you, is this nucleosome protected region. We saw that 140 onwards or more is potentially those nucleosome fragments, and anything less than 120 is the subnucleosome fragments. That's how we categorize when we trained our model. I mean, and now you can ask that why 120? It is a kind of after training ballpark number that we found out is that separates the subnucleosomal from the nucleosomes nicely. So that's how we came up with these two numbers at practical. And I have already described that how we got these overall study designs, like how we set this up. Is we started with this PDX. We have this whole perfectly pure human component, and then we have a paired tissue-derived knowledge side by side. And this is the kind of overall composition of the phenotypic subtypes who are epigenetically distinct from each other. So this was our start point. And from the start point, we also described that how we have actually learned from the tissue and gone into the real life patients and learned from that there are multiple features that we can extract from this cell-free DNA. So these are some of the features for now is the name is here. It's differential transcription by factor binding features or nucleosome depleted region specific features. Attack score, attack-seq open chromatin NDR signature score is the kind of second score we generated. That is another feature. Mean nucleosome phasing score is the third feature, and gene body transcription start site, that's another feature. And each of these features would be talking about different aspects of the biology, cancer biology, or transcription related biology in the data set. So that's what we will go through now. Let's start with this first feature, which is differential transcription factor binding site score, or we call it NDR score. So before going into it, what we have done is we take this entire PDX pool. I don't know what's happening now. Sorry. So we started with those PDX pool. We perform a whole transcript of analysis in duplicates. So that's how overall number of columns might be looking bigger than 25. So here is our AR low PC, NEPC, and then ARPC. We performed differential transcription factor expression profile, and we came up, there are 404 transcription factors that are significantly deregulated between NEPC and ARPC. And likewise, when we perform the total transcriptome comparisons between AR low PC and ARPC, that number is 110. So overall, total 514 transcription factor that are deregulated between these three subtypes. And before going into further details, I will highlight this particular transcription factor, ASCL1. This particular transcription factor is uniquely almost always upregulated in neuroendocrine prostate cancer, whereas AR or androgen receptor, this is almost always upregulated in adenocarcinoma or this ARPC or AR regulated prostate cancer. So starting with these two extremes would be the best option to start with, showing a kind of an example how we are using Cellfree DNA to show this epigenetic subtypes separation, like how we can capture the epigenetic subtype from the Cellfree DNA very uniquely. So what's happening here is we started with this kind of, so this is the cartoon which is showing how we can determine transcription factor binding site specific features from the Cellfree DNA. So the idea is when a transcription factor binds to a region where it's supposed to bind, the nucleosomes of that region would be depleted or it would be moved around, and all other nucleosomes would be kind of well organized, so it would become phased, and that's how it would create this peak and diff feature with this kind of giant NDR gap in between. And we know from our previous studies that some of these flanking, like transcription factor bindings, flanking nucleosomes get H3K27 acetylated in the process of kind of these reorganizations where the transcription factor would come and bind. And when the same transcription factor is not active in another tissue, it would not be that well organized. So those in those cases, the nucleosome would be much more randomly located. So if in one case, if we see these features in a particular transcription factor binding site, we know that this is exactly where that particular transcription factor came and showing its activity, whereas in the other case, it is not that, like here the transcription factor is not present. So to kind of, as a proof of principle, we started with this CTCF binding. CTCF is a ubiquitous DNA binding protein. It binds kind of tens of thousands of sites, more than tens of thousands of sites. So we focused on the 10,000 CTCF binding sites across the genome, and we try to capture how it looks like in Cellfree DNA binding sites. And we are showing here the nucleosome fragment specific analysis, that how those 10,000 CTCF binding sites are looking like. So here there is a giant dip that we are seeing, and then those phase nucleosomes across those dip, or NDR, is showing up over here. Whereas when we are analyzing with the subnucleosomal fragments, which is basically as the signature of CTCF binding in the same sites, it is creating a peak. Now, as I said, that CTCF binding is ubiquitous, so this binding is present in both NEPC and ARPC. So that's how we do not see any separations between these red and blue lines, because these red lines are from the neuroendocrine prostate cancer specific plasma samples, and ARPCs are from the neuroendocrine specific, adenocarcinoma specific plasma samples. But we do not see any difference. But what we are seeing here is those peak and dip features are nicely showing up. So then we go down further and start showing that what does this peak and dip features is practically meaning. So we started with these HTK27 acetyl profiles in the tissue samples. Remember, we started with the sample with the PDX lines. We collected the tissues as well as the plasma. Here we are showing those HTK27 acetyl profile from the tissues, and it is showing this in the ARPC. There is that NDR dip of AR binding sites. So this is the HTK27 acetyl profile for those AR binding regions where AR is supposed to bind, and that's what we are seeing is at the AR binding sites there is this giant NDR dip is showing up, whereas in NEPC that dip is not so prominent. So we could look at these regions, and we can infer that using this HTK27 acetyl profile, we can segregate ARPC from NEPC very accurately. And when we go down to plasma samples from the same set of samples, what we are seeing is at the AR binding sites, HTK27 acetyl like where we derive this HTK27 acetyl profile, we are seeing that AR specific giant dip again. And that dip is not present in NEPC, which is shown in red here. So just using this one single profile, we could segregate out ARPCs from the NEPC very robustly. So this is the AR binding site profile. Now we are focused on NEPC specific ASCL1 binding site specific profiles. And as we discussed, that wherever the transcription factor is active, we will have this NDR dip, and see this ASCL1 specific NDR dip is only present in NEPC, and that's how it has created this giant dip and the peak, and that dip and peak is not present in ARPC. And from the cell-free DNA, we are seeing the same thing shown up, that this NEPC specific giant dip is present here and not so prominent dip in ARPC, which is shown in blue. So just using these two particular kind of transcription factor binding marks, we could separate out NEPC and ARPC. But before going into that, we asked ourselves this question. There were 404 transcription factors that were differentially deregulated, and can we capture those 404 transcription factor binding in a similar manner? But in real life example, what we have seen is there was some transcription factor binding like AR, ACL1 and HOXB13, where the separations are nice in cell-free DNA, but there were several other transcription factors where transcription factor binding sites signatures were not so clean or not so informative for us. So what we found out is there were total 38 transcription factors. I don't know, it's actually cutting off here. There were total 38 transcription factors for which we have a very clear such separation of signatures like these peak and dip signatures present. So we use this feature to classify PDX plasma, ARPC and NEPC very accurately. So this was our first feature that we learned. Is it overtime? Which one? It is. So this was really an informative feature for us, and we could just use this one single feature to kind of separate out NEPC from ARPC. And then we go forward and we kind of performed ataxic analysis. We look for what are the ataxic binding sites that segregates NEPC from ARPC. We found out there are total 15,881 sites that are specific to ARPC, like those bindings are specific to ARPC, those dips. And there were around 11,000 such sites in ataxic samples that were, those dips are only present in NEPC. And when we actually queried those sites in Cellfree DNA sample, this is examples. This is exactly what we are seeing is this NEPC specific dip is present only in NEPC plasmas, not present in others, and vice versa. Like those ARPC peaks, dips are present only in ARPC's plasma samples derived from PDX and not present in other samples. So what we know is, we know that now in the genome, where to look at for these marks, and can we use this learning and apply it in a real life patient samples and derive the same accuracy to classify NEPC from ARPC? That was our question, and we will answer that in a few slides down the line. We also keep on asking this question that do we really need those 25,000 sites or 26,000 sites to query? Because that would be kind of sequencing the whole genome. And if we suggest clinicians to sequence a patient's whole genome, that is still, I would say, not viable options, even in the US. It's better to kind of have a targeted, more shorter panel. So to study that, what we did is we kind of ranked these ataxic binding sites based off of its p-value and pick up top 10,000s, and we perform the same analysis, and we have seen that just looking at those ataxic differential binding sites, can we separate this ARPC and NEPC accurately? And answer is yes. We go down further to 1,000 sites, the answer is yes. And then when we go down to 100 sites, it is still doing the classification, but it's really not 100% of the time we can accurately infer which one is ARPC or which one is NEPC. So our thought is roughly around 1,000 different AR binding sites is a kind of this number that is more practical. Like we can just query those 1,000 islands and design a targeted panel and use that for this classification purpose. So we have gone through the next, this is the third feature that I'm going to explain to you now, is this mean nucleosome phasing score, which is basically depends on this idea is when a gene is actively transcribing, there is a lot going on in that gene body. Nucleosomes are not evenly organized. There will be Pol2 passing on. There are some transcription factor passing on. There will be subnucleosomal subnucleosome formations to let the polymerase go or to have the chaperone activities goes on. So there will be lots of transient states would be created. But when the gene is not actively transcribing, all nucleosomes would be compact or would be organized in this case. So this is exactly what we are seeing is in the Cellfree DNA's overall coverage profile. In 93, HOXB13, this gene is not expressed in this sample. And here we are seeing this well-organized peak and dip features, much more compact, whereas in this 136, in this lookup PDX, or this is prostate cancer PDX, HOXB13 is highly expressed. And we started seeing those randomness showing up. I'm showing here just the one example, but this is universally true for many of the genes. Now, it is not true for the 100% of the genes, but only 30% of the genes. And now we have gone back and started querying what are some of the factors making some of these genes behaving like this and not the others. And one aspect is when DNA methylation is not involved in the transcription, like gene regulation. This is exactly when we started seeing these features more clearly, wherever the DNA methylation is involved in gene transcription, gene transcription silencing, we start losing those signatures. So we formalize this score, and just based off of this particular score, we asked that can we predict these genes expression deregulation between NEPC and ARPC. And when we run that, as I said, that only 10% of the genes it accurately works, and that's exactly what's shown up. These are some of the set of the genes where this particular feature actually have this ability to predict this gene expression very accurately, whereas the 90% of the other genes, it didn't show much of a promise at this point. So I will skip this particular slide because of the time, and I will move forward to the other feature. What is very exciting and very close to me is this, again, based off of this, using this Cellfree DNA feature to predict gene expressions or gene expression deregulations very accurately. We started naming it as I cannot name the features at this point for some reasons, but what we used is basically coefficient of variation. This is very easy approach of kind of that deriving how heterogeneous the fragment sizes are. Now the idea is same. If the gene is actively transcribing, then there will be a lot of activities going on. So there will be some nucleosomes bound to it. There will be some Pol2, like transient Pol2. There will be some subnucleosomal fragments that will be shown up in the gene body, and that would create heterogeneous fragment size in the gene body. And if the gene is not actively transcribing, then the gene would be much more compacted. So the only thing that would be there is nucleosome protections. So there will be only one kind of fragment size class, which would be more of a nucleosome fragment size class. So overall fragment sizes heterogeneity would be less. Just based off of this idea, we derive this one score, which is coefficient of variation score, which is basically standard deviation of fragment size and mean fragment size, but only in that gene body. And just using that feature, we derive this score, which is basically a coefficient of variant score, and we correlated that with gene expression deregulations or fold changes. You can call it as a fold changes here. And this correlation is pretty striking, which is basically positively correlated at a rho value of 0.844. And it's highly significant correlations as well. So just this how randomly distributed these fragments are in the gene body is basically predictor of overall gene expressions. Again, you can ask that whether this is universally true. Can we apply this approach to 100% of the 100% of the genes, like all 28,000 genes? No, it is true for 1,800 genes as of now, where we know that we can use this feature and predict. And many of these genes are basically active transcription factors or cancer-associated genes. So for our purpose, we can use this feature for at least a substantial number of genes and kind of derive their mRNA expression deregulations right from the cell-free DNA. Whereas those 90% of the cases where it didn't work, again, in those cases, some kind of DNA methylation was involved that basically regulates gene expression. So because you know that there are several genes that segregates NEPC, gene expression that segregates NEPC from ARPC. So we thought that can we just use these DNA fragments, fragment pattern profile or fragment CV scores to kind of classify NEPC from ARPC, and we applied that, and still we could separate out NEPCs from the ARPC very accurately. Now you can right? Because Anulapada is sitting right in the front, so he would definitely say that because these are your training sets, so you should definitely see this separation. So yes, I mean, we use these samples to train our model, and that's how we are seeing this such a clean separations from each other. But can we take these and apply it in the patient and validate it that it's actually happening in the patients? Can we accurately predict that? So to answer that, we did some kind of cross-validation. It's a computational approach to derive which model is working best. So we performed some cross-validation, and in that cross-validation, we found out that this open chromatin site in the ATAC-seq-based, like of these four, ATAC-seq-based approach is the best to perform the NEPC and ARPC classification, and you don't need anything else. It is best means it's 100% accurate almost. So we actually, for this particular paper or for this particular work, we stayed focused on only ATAC-seq based features and apply it in the real life patient samples. So there were 101 patients strong cohort that we derived. So these are from the patients. 101 samples were there. 21 of them were NEPC and 80 of them were ARPC. Our question was that can we take these ATAC-seq binding sites and infer who is NEPC and ARPC, not knowing them ahead of time. And when we applied our model was accurately inferred NEPCs and ARPC 96% of the times. You can, like AUC is 0.96, you can say roughly about 96% of the time our model was accurate in inferring which one is ARPC and which one is NEPC without failure.
This data is from actual patients, not from PDX mice.
This is not from PDX mice. This is from the actual patient. And here we have just 0.1x depth of coverage data for these patients. And still our approach worked very accurately. So this is another cohort. So this is the starting cohort, which consists of very clear-cut NEPC samples and very clear-cut ARPC samples. There were not much transient samples that are mixed phenotype and so on and so forth, which is kind of a staged sample, you can say, that was available here. UW cohort, which consists of 47 cases, these are much more real life kind of cases who just walks in the clinic. And in that cases, there were 26 clear-cut ARPC. Our model was very accurate in inferring those 26 ARPCs. There were 5 clear-cut NEPCs. Our model worked great. But then there were some mixed phenotypes who has multiple phenotypic subtypes coexisting together. And our model completely failed. Because our model was primarily trained to infer or segregate out ARPC, ARLOPC, and NEPC. But there were several other phenotypic subtypes. Now, those kind of cases, our model started failing. So this gives us one more reason and start thinking that how we define our model. This was basically a machine learning based approach that we adopted. And then we gone into a much more binary classification style here. We moved on to kind of more very common AI-based approaches, and still all our binary approaches has this limitation. So we realized that binary approach is not going to work. This is exactly where Robert is kind of make a key contribution, and he came up with a linear algebra-based approach. And instead of inferring yes, no, NEPC or ARPC or that kind of classification, so he gone into a different approach. That what percentage of this entire DNA pool is coming from NEPC, what percentage is coming from ARPC, and what percentage is from normal. So that gives us much more dynamic information from a single patient. So likewise, one example, so same set of UW cohort, if you see this case, it's basically saying that in this patient, 60% of this entire cell-free DNA is coming from tumor, which is adenocarcinoma, and 40% is coming from a normal, like rest of the normal parts. So this became much more informative, and now when we have a mixed phenotype, it's basically clearly saying what percentage of this mixed phenotype is NEPC, what percentage is ARPC, and what percentage is normal. That is exactly where the uniqueness came up, No other existing model with the cell-free DNA is doing this, and this has an outstanding application in the patients who are undergoing treatments. So some patients shown up, and we are giving a particular treatment, say that is specific for a androgen ARPC type. 3 months later, his AR shrinks and other subtypes grows. That could be captured in the next iteration of sample, and that's exactly what happened. I'm not showing up, but here in this particular pool, we have multiple time points from the same patients, and we have seen that along with the tumor sizes and tumor subphenotype, the clinical information that are available, our approaches are actually correlating well. At this point, we have actually signed up for a clinical trial where we would be clearly sampling patients with this goal, that what is happening over the time and can we capture that accurately using our approaches. So what we learned from this entire discussion is that nucleosome profile from cell-free DNA can accurately depict prostate cancer epigenetic subtypes or phenotypic subtypes. We call this as a phenotypic subtype because they are genetically same, but they are phenotypically they are different, and also expressions are epigenetically they are different. ctDNA driver AR activity signatures can be a clinical determinant for standard of care anti-androgen therapy. So this is one thing, that whether AR binding sites are captured by AR, that itself is informative that whether somebody should keep on getting anti-androgen therapy or not. So if I'm taking a sample from day 1 in a patient and maybe 3 months later, if we sample the same patients and we don't see that its AR binding is occurring, like AR bindings are gone, we can stop giving that patient anti-androgen therapy and start thinking of the next iteration of treatment which is more relevant. Then ctDNA CV signature can accurately predict differential expressions of several genes that are clinically relevant. As I said, that 1,700 is the number of genes where this prediction works very accurately. For others, it is very leaky, so we would not claim that it is working there. But we are actively working to find out the newer features that is giving us more accurate hold on this. Actually, not the features. I mean, we are kind of working with the math, like different tweaks, like how we can capture the information there, present there. And it is, I think, potential tool for precision medicine, especially for longitudinal capturing the longitudinal progress of the disease. One challenge is still there. That the challenge is, if the patient is responding, when the tumor volumes are low, the overall tumor DNA contents are started becoming lower. At that point, this approach starts showing lower sensitivity. What we have found out, 8% is the sweet spot. 8% tumor DNA content is the sweet spot, above which this method works very robustly. And 3% is the lower limit, below which we would not claim that we can do any prediction. 3 to 8, overall performance is lower, but still this approach is usable. In that 3 to 8 range, I would not use this Keraun approach that Robert has developed. At that range, probably this particular approach, the binary classification approach, works best. If it is greater than 8%, this Keraun approach is great. So we have worked that up as well. So we are taking this in our account, and as we go forward and we have a larger number of samples, we will definitely also do that test, that how good we are doing. Or there are current approach somebody in Boston is working on, is giving a chemical dope to a patient few hours before collecting cell-free DNA. It is basically anti-DNase dope, so which would boost overall ctDNA content temporarily to a patient. So that may be the kind of a next generation iterations, that how this would be becoming more usable as we go forward. And definitely it is a potential companion diagnostic tools. It can be a potential companion diagnostic tools where that's the new therapies coming, or epigenetic inhibitors are coming. We know that how we can kind of do the testing and decide that whether to kind of keep on giving this treatment or not. With that, this work is not complete without the active participations of Gavin Hart, Robert Patton, Peter Nelsons, Brian Hanratty, and many more. So this is not a single person's task. It was really a big teamwork, and this teamwork is really worked out well. And I will end. My apologies. I mean, as I transfer my slides, it has changed some of the fonts, and also this alignment. I hate to see this one. It's like looking like this for dear Shoda, and all the contributions he has to all of us who were there during ISI days, and I really miss him. With that, thank you. And I'm still actively hiring for my lab. I'm actively hiring a bioinformatician in my lab. So if anybody is there, you can recommend, please go ahead, and I will definitely go for it. And we are coming up with a new cancer center building over there, which would be, our lab would be moving to this new one in September. It was like this, and by now, they already has the first story built, so it's rapidly progressing, and by 2024 we'll be there. And happy to take questions.
Any questions? I have one question.
Oh, absolutely.
I don't need that. Oh, okay. Hi. So these 128 of patients or so you showed. Did they have also metastasis?
They are all metastasis cases.
So in that case, would you be able to know what is the location of the NEPC?
No, that's the challenge part. Is we can right now tell that among the entire pool, it's a one single sample for the entire tumor sets that are present. It is not tumor specific. That was the challenge. Like, this is exactly what we wanted to avoid. If there are multiple tissues, we cannot do multiple biopsies. But this is a kind of global summary of this all the tissue pieces that are there.
Okay. So now this brings me to the next question. Could you, yeah, here this was a convenient sampling and continuously sample the cases as they came.
Longitudinal sampling, you mean?
No, what I mean is that this patient selection, was there any criteria or continuously kept on?
So these are the patients that are regularly coming to the clinic, yeah. And as a part of a trial, they are keep on collecting 10 ml of plasma from them.
Yeah.
10 ml of blood.
So in general, what is the percentage of mixed phenotype in the cases that you see in your center?
Okay. So here is the point: is whether these patients are responding to androgen deprivation therapy in terms of PSA. Based off of that, we know that these patients are ARPC. And based off of that, we selected this other set where we know that whether neuroendocrine phenotypes are present or not clinically, or the mixed phenotypes are not. So yes, I mean, you are right. This overall mixed phenotype wise, this is kind of it's not a representative. It's more than that.
So mixed phenotype will be more than.
No, mixed phenotype would be little less than this.
Okay.
So we intentionally put that because it is a challenging group. And we needed to learn more about these subtypes to kind of come up with a future solution, this is one, but we're still not happy. When we have to come up with, there are a few other phenotypic subtypes. That is, we are still working on, we are kind of looking for the newer PDX models with these mixed phenotypes. So there is an Australian group who has actually generated this mixed phenotype PDX model. So they are shipping the blood plasma to my lab very recently.
So in terms of metastasis, NEPC usually does not develop locally.
It like very rarely, but extremely rarely.
Okay. Right.
Yes.
Wonderful talk.
Oh, thank you.
My question is, are these patients from where the xenografts have been prepared, they were treatment naive? Or do you find, or do you see any effect of pre-treatment on the different kinds of cell-free DNA profile?
No, that's a great question. So actually, these PDXs are not treated, but the original patients where this tumor was collected, it's a mixed bag there. But what we are looking at is AR binding profiles where AR binds. And we are also doing the tissue-based analysis. So our job was to see side by side how good they are representing well. And as an overall bag of all the AR pieces, whether the entire AR bindings are represented in Cellfree DNA or not. So we were not kind of focused on one single PDX, what's happening. That's why we needed more.
Okay, I kind of understood what you're saying. My second question is regarding this mixed profile cases.
Yeah.
So essentially, these patients were diagnosed with a single type of cancer, but you are getting mixed profile from the Cellfree DNA.
No, so these are the patients, I would say, that they walked into the clinic as a probably adenocarcinoma, and they are keep on under treatments for years. And as a time point onwards, they stopped responding to androgen deprivation therapy. That is the standard of care. Then they become CRPC, or castration-resistant prostate cancer. And people keep on studying them, and they have some clinical profiling. And based off of that profiling, they already knew that they have a mixed phenotype clinical features. They are not accurate. The mixed phenotype, we are saying mixed phenotype, but they are not accurate. Still not accurate. We cannot say what percentage of this mixed phenotype is true, because the clinical data itself is not specific or sensitive enough.
Yeah, I can understand the problem. So in that case, do you find that the time of the treatment for this cohort could affect?
No, it is not, because there are few patients who are really aggressive. Their overall response time start to end maybe less than a year. Whereas there are a bunch of other patients who are doing it fine for 14 years, 15 years, and then developing more aggressive phenotype. So the heterogeneity already exists in the sample profile itself.
This clinical phenotype, what does this mean? Clinical phenotype of the mixed individuals?
It's based on sometime histopathology, some GRD.
On the top, there is something which says about the clinical phenotype.
So it's absolutely, I would say, subjective clinical phenotype. It's not a very quantitative approach.
There's a new color, introduction of the new color in the clinical phenotype.
This gray color, that means really gray.
Navonil.
Yes.
So, I saw somewhat K+2 in your slide.
Yeah, absolutely.
So, can you just go back?
You have seen it, but 10+2 is definitely more.
My question is whether have you detected 10+2 ERG fusions?
Yeah, very easily.
ctDNA profiles.
So detecting fusion is easy with Cellfree DNA. Standard analysis.
Do you have any signal?
From ctDNA, you can see those 10+2 arc fusions like those.
Yeah.
So it's basically show like a shallow deletion between these two genes, 199 megabase apart.
So
Sorry, kilobase apart.
So you also detected that from your samples?
Yeah, I mean there are some there. But that doesn't mean that we can use those to predict the 10+2's overall expression that is dependent on ARC or without ARC. So these are some studies that are ongoing is when we are detecting this deletion along with this kind of gene expression signatures, can we use these as a prior to train our model more kind of gene by gene approach?
So what percentage of your samples have these 10+2 ERG fusion?
A training set or
No, in the
clinical set?
Validation cohort.
That I cannot say. I don't have the stats. In training sets, yes, I mean training sets, there are 30% of them has 10+2 arc fusion. And that's the natural frequency.
Anyone else?
Anybody in the online YouTube has any question?
All right, so thank you, Navonil. Thank you for the great talk. So he's here for a few more hours, so you can grab him if you have another question, or if you can grab him
Yeah, I mean, I agree that there was a lots of new concepts, lots of maths, subtypes. There might be something confusing. What some of you might find it confusing, that what is NEPC, ARPC, and here and there several times. So please feel free to shoot me questions, even if it's naive. No question is naive. Maybe it would give us some new thoughts to kind of work more. So please come up and throw us a question, throw me a question. Yeah, that's an option. So for diagnostic purpose means whether somebody has cancer or not, that's very easy.
No. So if somebody has a cancer, then for diagnostic purpose, I want to know that.
Hello.